And so hello, everyone, and thanks for joining. Thanks, Yaxal, for accepting our invitation. So Yaxal did his PhD in microbial ecology at the Department of Evolutionary Ecology of the Institute of Ecology of the National Autonomous University of Mexico. And after finishing his PhD, he joined as a postdoc, the bioinformatics group at the Institute of Bio and Geoscience at, I'm gonna see if I pronounce it correctly, but sorry if there is any German here, at the Forschungszentrum Jülich to work on plant genomics. And after this, he joined the microbial paleogenomics unit at the Institut Pasteur. And now he focuses on the analysis of ancient oral microbiome. And today he's presenting ourselves to all of us his work. So thank you, Yaxal.
Bianca. Well, today I will present you one of the projects in which I have been involved. This is already published, so yeah, I mean, there are not many changes that we can do now. But okay, the title of the manuscript was an ancient Epstein Barr virus genome record from a museum's Penisgar from Papua. Wait, sorry. Okay, so this is the case. It was published in this journal, the Journal of Infectious Diseases, and was a working collaboration with Anais Ukias and Philip Charrier from the laboratory of University of Anthropology. Yeah, well, so well, I will start first with what is an Epstein-Barr virus. This is one of the this is a member of the human gamma herpes virus family, and was first isolated of the virus from the Burkitt's lymphoma. And what is very interesting about this virus is that normally it is transmitted within the close family via salivary or during the infancy or childhood. And it infects, depending on the sources that you look at, between 90 and 95% of the whole adult population worldwide. So it's a virus that is really well distributed all around the world. This particular virus has two different subtypes. The type 1, that is normally ABV1, that is ubiquitous worldwide. You can find it, for example, in Western countries, East Asia, Africa. And the second type is the type 2, that is mostly prevalent in Central Africa and New Guinea. Overall, both the subtypes have a very similar genomes, but they can be differentiated mostly based on the EPNA genes. That these play a role mostly in the viral persistence, the transformation, regulation, as well as the expression of the proteins of the virus. Another really nice thing about this particular virus is that it has a really congruent geography phylogenetic. For example, this division has been observed all around the world. You can see in several worlds. For example, you can see there are some clusters in which you can group mostly individuals coming from Europe, some others from Asia, from Africa, from Oceania. And you can go even more in detail and find inside this geographic region, this geographic regions, more like a fellow population. For example, we have in Asia some that are associated also from different kind of tumor that they form. For example, we have this Epstein-Barr population 2, 3. They are this is a very well structured virus in general. So this is a really nice thing about the virus that gives you this information. Now, on the other side, what is a penis guard or a coteca? These are traditional clothing worn mostly, well, basically, by native male inhabitants from highland ethnic groups in New Guinea. And normally it is made from this calabash, this dried out guard, and the shape and the angle of the coteca is also associated with the social status of the person who is wearing it. And I mean, in the Museo du Quebra link from the Yaxil collection, which is the one from which we obtain the artifacts, they have a collection of them. So 19 of them were a sample on the surface, And this came from different locations, from Papua New Guinea, from Vanuatu, Indonesia, but also they have like a different materials that like in which they were made. We have like a typical one from calabash. In some cases were like more open, some were closed. You have like this round, like a long curve, so like we have like a different kind of materials. So some of them were from this hornbill beak, and the time periods were from the like late 19th to end of the 20th century. So they are not that old, but they were like the ones that they were available at the time at the museum. And just like to show like the hornbill, what is this? It's like this bird. So they have like this coteca made from the beak of one of these birds. So once that we have like the artifacts, for the sample was like the normal like a kind of normal procedure. So it was like we have the sampling from the surface, we have the DNA extraction, DNA quantification. But one important thing for this particular project was the original aim of the project was to analyze human DNA. So that is why there was a step in which it was like targeted, and the selection of the samples was based exactly on the prevalence and the quality of the human DNA. So based on this on the selection, we kept 13 of these cards, and they were the ones that were selected. Then, for historical reasons of the project, things happened, and we decided to focus on the Epstein-Barr virus. So when we started looking at them, so these are the 13 samples that were selected, and only one of them, this one that we say in 1998, we identified like a good amount of rates that were targeting the Epstein-Barr virus. For the rest of them, we had like very few rates to be able like, to consider like positive selection. So we didn't move forward with them, and we kept only this only sample. And the one that was selected was precisely the one from the hornbill beak. So for this particular one, this was dated between 1950 and 1970. We don't have like exact date for this particular artifact. And as we didn't know which kind of the virus we have, we performed all the analysis based on the two types, the type 1 for this reference, B95, and the type 2, the AE876. So we did everything kind of in parallel. But we wanted to be sure at first that the data that we were recovering were kind of ancient or were contaminated. So sadly, in this case, we didn't observe like a clear pattern of contamination. There's like a small thing only in the first base, but we think that this could be because of the recent nature of the artifact. So it's not at most like, we say like between 1950, 1970. So 1750 years is not that old. That maybe that is why we are not able to recover like a more clear pattern of domination. So, but we decided to keep moving forward to see if we could get like something. Then what we did was like to try to get some public information that was already out there. So we collected 238 public genomes from this virus. And also we decided to try to recover more of the virus from some oral genomes that were already like there. So at the end of doing like a screening, we kept only 237 genomes. We removed one because of quality, and we could recover 4 metagenomes, including ours, that were positive to the Epstein-Barr virus. At the end, what we classified more or less were like 212 that belong to the type 1, what were classified as type 1, 22 as type 2, 2 that are classified as a recombinant Epstein-Barr virus, and our strain that we didn't know which type it was. And our like representation from the geography that we have, they belong in general like to 27 countries. So because we have like a wide distribution worldwide to be able to see also like to look for these global patterns. But the first thing that we wanted to do was to issue like which kind of which type of virus we were looking at. So what we did was like to map against the reference of the type 1 and the reference of the type 2, and this is what I am showing here. So on this side, what you are looking at are all the Epstein-Barr virus that were classified already as type 1, mapping to the reference of type 1. In the second one, the green one, we have the type 2 mapping to the reference of type 1. The third one is like all the genomes from the type 1 classified as type 1 mapped to the reference of type 2, and type 2 mapped to reference of type 2. So basically what you can see is that when we map as expected, the genome of classified as one type to the one that they corresponds to itself, they have like a higher mappability. And the blue dot in these two measuring plots represent our sample. So you can see based on this that we have a higher probability to the type 1. So this is like the first indication that the virus that we recover belongs to this particular type, Epstein-Barr virus type 1. Then we also decided to evaluate like the rate of coverage, which is like the percentage of the genome that is covered at least once during the mapping. As well as before, we were mapping against reference of type 1 and reference of type 2 based on the book classifications. And again, what we saw was that the virus classified as type 1 had like a higher rate of coverage when they mapped again to their own reference than when they were mapped to the reference of the type 2 and vice versa. And again, in this case, we have that our sample, the one that is in this case in red here, has like a higher rate of coverage when we are mapping it to the reference of type 1 that compared to the reference of type 2. So again, this gives like a support that that this belongs to the type 1. When we look at more into detail, for example, we saw that the rate of coverage, well, in this case for type 1, was around the 80%. So we are covering around 80% of the genome, and in this case, it was around 75%. So yeah, there was like a difference. And as I said before, one of the features that I use normally to be able to classify the Epstein-Barr are the EPNA genes. So we also decide to look into them. And this is what I am showing you here. So what I am showing is like the windows of the different EPNA genes. We have the EPNA 1, 2, 3A, 3B, and 3C. On top, we have our sample, 1998. Then we have one representative genome from type 1 and one representative genome from the Epstein-Barr virus type 2. And this is when we are mapping to the type 1. What we can see here is like for the type 1, we have like a very high coverage of all the genes. For the type 2, like a bad coverage in general, except for this one. And for the type 2, and for our sample, it's kind of tricky because it's like we have like a dot, like a mapping that is not really consistent, but it's like up and down. But when we compare this, when we map to the reference of the type 2, we can see that there's like a real difference, so that the mapping is much better, we can say like that. For example, in this case, we have a peak of region going up to 30, but on average, we are around a coverage of 10x, while for the type 2, basically, I mean, the highest peaks are around 15, so it's like still like a way lower of what we're expecting, but we have genes that really are not mapping at all. And this pattern is more similar to the one that we observed for a virus that was already identified as type 1 compared to the type 2. So this, I think that with all these tests that we made, it's like we can say that basically this strain is most likely an Epstein-Barr virus from the type 1. Then we decided to move forward and see about how recombinant this virus could be, because as I mentioned before, there are some cases in which also there are some cases that are recombinant. So this is also the first like a PCA in which we are observing different things. So what we observed here was like the first and the second components allow us to differentiate basically on the geography. Here we have a cluster that can be classified basically from all the individuals coming from Indonesia and Papua New Guinea. We have here in circle in purple, mostly individuals that come from East Asia. And in this blue circle, we have like the rest of the world, basically, that is Africa, Europe, in general. That if you remember, if I go like back, you have like this big cluster. Sorry, I think it's like here. Like here. So we have like this big cluster from Asia. We have our cluster from Indonesia, Papua New Guinea, and the rest of the world is kind of mixed together. So it's kind of similar to what we are observing in this plot: Indonesia, Papua New Guinea, East Asia, and the rest of the world. And then when we look at the components 2 and 3, this helps us to differentiate basically between the Epstein Barr type 1, that everything's on top, and the type 2 and the recombinant that are here. So in this case, the recombinants are these two in squares. And our individual is falling in this part. So again, it's falling where we expected based on the previous result. This is when we look at the type 1. Now, if we do the same based on the reference of the type 2, basically we get similar results. We have, when we look at the components 1 and 2, we have our group of Indonesia, Papua New Guinea. We have our sample falling in Indonesia, Papua New Guinea. We have another one from East Asia, and we have the rest of the world. Now, when we look into the components 2 and 3, exactly the same. We have one group, one group basically with the type 2 and the recombinants that are again here and here. And we have the type 1, and where our strain is falling in this part. So yeah, I think that this is also like a good support to say that India really is like a type 1 strain. So finally, what we were missing to do and we decided to do was to analyze at a phylogenetic level. For this, I mean, we make like a phylogenetic reconstruction of all the genomes that we analyzed. And what you can observe here first, we have one group that corresponds basically again to the type 2, together with the recombinants, the ones that are here in purple. And as has been reported before, it consists mostly or is predominant in individuals from Africa and New Guinea, while type 1 corresponds again like is the rest of the world, and we have like a big group of East Asia. That is what we observe here. So again, we have like our big cluster of East Asia and our group of Indonesia, Papua New Guinea, where our sample is falling exactly in the cluster of New Guinea. That if we make a kind of a zoom in, we have it here that is kind of basal to this particular branch, in which we have most of the individuals that were isolated from a person from the Papua New Guinea. So I mean, this is what we did. So basically, I think like to conclude, what we can say is that we recover an Epstein Barr virus from a museum artifact. There are previous reports of this virus in the chewed birch pitch, but it was recovered from an excavation and was the detection was based on 593 reads. But I think that until like, we know it's like the first report from the report from an artifact that has been maybe hundred and exposed like for several decades. So this is like the difference of this particular study. We were also able like to determine the type of the strain of the Epstein Barr virus. And what was nice was that it was this congruence between the muscular information regarding the depth of the origin with the genetic information of the virus, which will help us to kind of like rule out the possible contamination, and also it shows also gives like a support that the museum artifacts can also conserve like these genetic records and they can be used indeed to as an object, like to try to get new information from them instead of or trying to reduce the current methods that are like more destructive of the biological samples. Yeah, and with this, yeah, it's thanks. So if there are any questions or anything.
So I have one question. In the meantime, about so I well in reality too. So the first one is about the you spoke about the non-destruction of biological samples, and I was curious. I don't know if you know, but how was the sample taken?
Yes, and for the sampling, what they did was like they took Q-tips and they basically just like took it from the surface.
Ah,
So
okay.
from what I understood, there were like two different methods of doing it. They tested with like dry Q-tips, but also with with wet Q-tips with water.
Mhm.
And at the end, the ones that were conserved were the ones with dry Q-tip sampling. So that's the way they did it.
Cool. And also about the phylogenetic tree, um, how like is it a neighbor joining tree or which kind? How did you perform the phylogeny?
Yes, for this, like, we use like we was like like the prep process of the tree was with the like pipeline that we have like at the lab. That way, basically, what we are doing is like we use the reference genome, and then we place on top of the reference genome the SNPs that we detect.
Mhm.
We keep the genes that are present in more of the 50% of the samples. If one gene is missing more than 50% of the samples, we remove them, and the rest we keep them. For this case, the final alignment was around 150,000 base pairs from the 1072 that is the genome of the sex of the genome. So we kept most of the genome, and then it was a maximum likelihood.
Okay, thank you. And
Eh, Yeah, go ahead.
no, yeah, I was reading there is
The
a question
online,
in the
yeah.
chat. Yes, from Charlotte. She says, interesting talk, thank you. Is there much modern data on ABV viral load in genital tissues? Did the wearer have an EBV plus cancer?
I mean, in this particular case, for the samples that we have, we didn't have more information regarding the origin of the artifact. Basically, that they were collected in the between end of the 19th centuries during the 20th century, and that's it. We didn't have like any record of the clinical pathology of individuals because it was more for the artifacts, not the base for there was not a clinical interest for the collection of the artifacts, basically. So in that case, we don't really know if there was like a positive case of the Epstein Barr or there some kind of cancer on this one.
Hi, yourself. Really nice talk. I wonder, like, because given we know Epstein-Barr's virus is like a highly recombinant genomes, so I wonder, did you consider like removing those potential recombinant sites and then we build a tree? Will shift a lot for the tree topology compared to what you have now.
I mean, we consider at some point that because of us, well, it was not kind of proposed by the reviewer, but they were like a concern also as well from the combination. So basically, at the end, what we did was like to run this was RDP5 to test like for the recombination, and it was, I mean, there were like yeah, for we got like we got the signal for recombination for a couple of samples, but they were not significant for most of them. So at the end, we decided to keep them. But yes, I mean, it's true that these regions can be like very recommended and can modify the topology, but at the end, we didn't test that.
Okay. Because I did similar stuff on our ancient EVV study,
And it changed a lot.
but then we observe a huge change within the tree topology if we remove all the detect recombination sites within the genome. So I wonder whether you will suggest
How
we should.
would do? I mean, I think that, I mean, the problem is that most of the information that I think it's out there is you don't see that most people remove these regions. So, I mean, that's the problem, that if, I mean, let's say we can kind of divide into two parts. If you remove the recombination region, because exactly you know that this they can affect the topology, they will kind of, as you have already seen, change a lot to what is already published or known, and you need, I think that you will need, in this case, methodologically, I think it's correct, but you have to have now more support or more data to be able to state why you are observing that I mean that fact basically, no? So it's, I mean, it's hard to say which is the right and it's the right way.
Yeah.
But yeah, I mean that's the problem always with the combination, that depending if you consider it or not is what you observe at the end.
Okay. Thank you.
Are there any other question for Yaxal?
Can I have one more question?
Yeah.
So
Many as you want.
regarding the PCA result, are you using the biallele SNPs, or you also including those multi-allele SNPs you called?
Again, sorry. Can you
So
repeat that?
are you only using the biallele SNPs? Did you do any filtering for the
Yes,
SNPs?
yes. We
Or
get only the VIL SNPs. We remove the others.
because I also do exactly similar stuff. I map everything against to the type 1 reference. And for my PCA plot, the PC1 are mainly separated by the type.
Okay.
And then,
I mean, in my case, yes, we also remove all the SNPs that have several variants, several alleles, and that's how we build it.
okay. So did you also do any potential filtering to sort?
Yes, I
So
mean,
you including all the biallele SNPs without any
yes.
further down filtering.
Yes, we are keeping the via positions. That's the word that we remove. In that way, that we yeah.
So I have a follow-up question. So we do similar stuff, and then I realized that it's quite easier to have a reference buyer if we're using reference-based method to construct the genomes. So one of our ancient samples, actually, if I map to the type 1 reference genome
Mhm.
and then call the SNPs, they will falling on the position of between the type 1 and type 2. But if I map to the type 2 reference genome,
Yeah.
that specific sample are exactly perfect, locating on the type 2 cluster.
Okay.
Yeah, but then I'm manually
Mhm.
checking on those two hypervariable regions. That's the EBNA2 and EBNA3.
Mhm. Mhm.
I see exactly the similar pattern you testing on your supplementary figure. So for,
Which
I think you're not showing maybe further down.
probably not. I mean, do you remember which figure?
Yeah, this one. I'm
Okay.
doing exactly similar stuff you
Uh-huh.
have here. And I observe exactly the pattern like the bottom row.
Ah,
So I assume it should be more close to the type 2, but I don't know why when I map against back to the type 1, they will,
you also have like
yeah, got it on the between the type 1 and type 2.
yeah. Yeah, because I mean, in our case, it's, I mean, for all the tests that we made, it seems like to be really falling more like in the type 1. We don't see like the commit like in this intermediate state. Could it be? I don't know. Like, so,
But then I run the recombination detection, the RDP5, and then
uh-huh.
actually I don't observe too much recombination event
Yeah.
happen on that genome. So I wonder how,
How the mapping
how
could be
why
kind
we
of
are,
guiding.
yeah. But you have a pretty clearly structure, like should be more close to the type 1 ABN
Yeah, I mean,
strains
I
on
guess
your
it
case,
seems to
right?
be, in my case, it seems like to be really like more close to the type 1. But I don't know, I'm not examining for the mapping, is the could be the only way if I don't know if the mapping parameters are like a bit relaxed so that it allows to map more to both references, and that is why you are kind of observing the
Mm,
like the intermediate state.
okay. I think I just using quite less mapping steps. I allow one mismatch
Okay.
and then give a less penalty for the like gap penalty. So I allow more mismatch or
Yeah, that's what I would maybe think that's like
because I read your
the
paper, you're
parameters.
using the BWA.
Yeah, exactly. Exactly, that's
Yeah.
what I was using.
Mm.
Yeah, I mean, like what you're describing, yeah, it's kind of difficult to distinguish like, what is going on.
Mm.
Could it be co-infection, maybe? Of two. Can it happen? I don't know.
I'm not sure honestly about the confections. I haven't read a lot about that. But
So you mean so did
Oh,
Mario propose both
yeah.
of these type
Mm.
co-infect with that particular people?
Yeah, I don't know.
I haven't heard this, but it's kind of like quite interesting. Um,
Okay, I think if there are no more questions, maybe Joao, you can contact better Yaxal so you can maybe discuss it and see if he can help in this. I will stop recording and I will wait, stop recording first.